On behalf of Cambridge Innovation Institute's Global Web Symposia Series and our sponsors, Genovac, ENPICOM, and Carterra, I'd like to welcome you to Discovery, Characterization, and Generative Deep Learning-Based Humanization of Therapeutic Antibody Candidates. My name is Elizabeth Lamb, and I'm the host and moderator for this event.
Okay, thank you, Elizabeth, for the kind introduction. Yeah, so as Elizabeth mentioned, my name is Andreas Weise, and I'm working as an account manager at Genovac. And in this joint webinar, we will talk about the discovery, characterization, and humanization of anti-RAGE antibodies from multiple species. So the webinar is basically split into two parts. So in the first part, I will give a short introduction about Genovac's capabilities and services, and then I will talk about the discovery production of the anti-RAGE antibodies. And a major point will also be the characterization of the anti-RAGE antibodies using the Carterra LSA high-throughput SPR platform. And then I will hand over to Piotr from ENPICOM, and he will talk about better humanization tools, and and he will report on how we use the GenovacAI humanization of anti-RAGE antibodies powered by the ENPICOM tools. Okay, let's start with a short introduction to our company. So Genovac works as a CRO CMO with facilities in Freiburg, Germany, and in Fargo, North Dakota. And our services are basically split into main services. On the one hand, we have the antibody discovery services, where we apply multiple immunization technologies, can apply multiple species, and we have also multiple screening options, including, of course, hybridoma workflows, but also we offer multiple single B-cell screening platforms, and we have also the option for hit expansions using repertoire sequencing using our NGS facilities. And on the other hand, we have the production and characterization part, where we have lots of options to go for different scale of antibody production. Then we do humanization of antibodies, and we can also characterize antibodies, for example, doing antibody binding kinetics and epitope binding. And apart from that, we have also other production facilities or capabilities as indicated in the last bullet points. So Genovac was founded in 1999, so we have now 25th anniversary, so we have a lot of experience. And within the last 25 years, we have completed more than 3,700 projects. And amongst those, there were a lot of challenging targets and to which we were able to generate antibodies against. Our core experience is the genetic immunization. And with the genetic immunization, we are able to discover highly specific antibodies generating native epitopes, which is a prerequisite for therapeutic antibodies, for example. Concerning host species, we are quite agnostic. So we can immunize and have a lot of experience for the immunization of the rodents, including all kinds of different transgenic platforms. But we also regularly immunize rabbits, camelids, and human, or we don't immunize humans, but we have access, we can screen human-derived PBMCs. So, and we have taken a huge focus on adapting all these different animal platforms to our single B-cell platforms, including the Beacon screening. For the antigen formats, by our long experience, we have made the experience that very often genetic immunization is sufficient and very often, the best option if the intention is to generate therapeutic antibodies. But we have also a lot of experience using different antigen formats. And this, of course, also depends on the objective of the different antibodies. So, for example, for tool antibodies or diagnostic antibodies, we would at least include also protein immunization, for example. And for very, very challenging targets, we have also alternative options such as immunization with AAVs or VLPs or cells, for example. And yeah, as I just mentioned, by our experience, we learned that for most of our targets, the genetic immunization turns out to be the best option, as it has some really good advantages if you compare this to other antigen formats. First one, and not unimportant, is it's a very economic and quick and fast method to generate the antigen, because it's basically molecular biology approaches. Other advantages is, of course, that you have a great flexibility in the antigen design, so you can generate different immunization constructs. So, for example, to direct the immune response to a certain epitope of the target. And so another good thing is that the genetic immunization leads to the fact that the immunized host is producing the antigen itself. So this is really native epitopes, and this is, of course, important if you develop therapeutic antibodies. And we have learned that by genetic immunization, we frequently discover really high affinity antibodies. Okay, that's enough of the general introduction. And with this, I would like to move on to the discovery campaign of the anti-RAGE antibodies, which we will talk about in the next half an hour. So we started this collaboration on this target with the Leclerc Vetter Research Lab from the North Dakota State University. And they are working on this target since a long time, so 15 years. And they have tried different approaches to generate highly specific and high affinity antibodies by using hybridoma and phage display technologies, but they did not succeed. And at that point, they contacted Genovac, and so we ended up in a commercial collaboration for the discovery of therapeutic and diagnostic anti-RAGE antibodies. Some words about the targets. So the receptor for advanced glycation end products contributes to several human diseases, as it is involved in a broad range of different cell signaling processes. And it also relates to quite a lot of certain serious diseases, such as Alzheimer or different cancer types. And what you see here is the membrane-bound form of the target. There's also a soluble form, but in this campaign, the target, and we focused on the membrane-bound version of the protein. And as you can see by structure, it is a typical type 1 membrane protein. So it has a cytosolic C-terminal domain, then it has a transmembrane domain, and then it has a extracellular domain. And this extracellular domain is composed of 3 main subdomains. And the terminus, we have a variable domain, which is then followed by 2 constant domains. And for completeness, we have also a membrane proximal domain. The mode of action of the protein is that as it binds a ligand, and there's a different range of several different ligands, the receptor builds homodimers and activates downstream intracellular signaling pathways in a positive feedback loop manner. And this is this is somehow serious if it is upregulated too much, because then it just keeps on promoting RAGE expression and RAGE signaling in a feedback loop. So the primary aim is to generate antibodies that bind to the variable domain and thereby to blocking the RAGE activator binding and therefore interrupting the signaling of the RAGE molecule. So to ensure that we will end up in a successful campaign, we choose a strategy where we have immunized multiple species. So finally, we came up and immunized mice, rats, and rabbits. And this is just quickly the immunization for the mice. So the primary objective for those antibodies is that we wanted to generate therapeutic antibodies. So we selected genetic immunization at the antigen format, and we used two different wild type mouse strains. And what you see here down is the Sera test results. And as you can see, we had quite variety in the response levels of the different animals. But there were also a couple of animals which showed really very strong immune activity if you compare this, for example, to the control antibody, which is also shown in this graph. For the rats, we used only one cohort, including 3 wild-type rats. The objective is more or less the same, so we aim for therapeutic antibodies. And what you can see is that on average, the immune response was rather low, at least in comparison to the control antibody and to the mice. However, this still should be sufficient to find or to successfully screen for antibodies with these animals. And finally, for the rabbits, the primary objective was to develop diagnostic antibody candidates. And therefore, we had 2 different cohorts. And 2 rabbits we immunized by protein, and the other 2 rabbits we immunized with DNA. And as you can see, also with the rabbits, irrespective of the antigen format, we reached quite good immune responses, as shown here by the FAX data for the protein group, or by ELISA data for the immunization group, protein immunization group. And the reactivity levels of the genetic cohorts, at least on the FAX based data, is very similar to the protein cohorts. Okay, so we had all the animals showing more or less a good to very good immune response. And then we choose as the screening platform the Bruker single B cell platform. And in general, the Beacon provides us with some advantages above other screening methods, and we profit a lot from that. One example of advantage, is that you can simultaneously screen different animals, even immunized with different formats simultaneously on the Beacon. Then we have also developed improved culture media to extend the B cell life. And the other advantage is that you can run sequential assays on the Beacon, which are very highly sensitive. And this allows already on-chip profiling or down selection of hits if necessary, if you have a lot of hits. And concerning capacity, with our 2 beacons, which are assigned for B cell discovery, we can screen up to 160,000 cells if we use the 2 machines on 2 consecutive days, and we can export up to 768 specific antibody or specific antibody producing B cells. And if we then would include also the repertoire sequences or hit expansions hit expansion options we have, this enables us to do million cell screenings. For the RAGE campaign, in general, we use the plasma cell workflow for the rodents, for the mice and the rats, and for the rabbits, we use the memory B cell workflow. So because we don't have the right tool antibodies to isolate the plasma B cells, which are just not available. So for the rabbits, we have an initial activation step of the memory B cells, and then the activated memory B cells are loaded on the Beacon. Then we have the 1 or 2 day screening on the Beacon, and following that, we move on to the downstream high throughput cloning and sequencing, and then we can further characterize the antibodies. And this table just quickly summarizes the results we got from this screening. And as you see, we got quite variable numbers of hits on the beacon. So we got lots of hits from the rabbits, a good number of hits from the mice, but more or less a relatively low number from the rats. So this was a bit surprising. But as you recall, we had, on the one hand, we had the lowest immune responses in the rats. However, this maybe still should have been sufficient to maybe get more hits, but this is biology, we think also, and so we were happy that we used a multi-species approach, because you never know it in advance with which species you end up really with a high good hit number in the end. So for the further downstream characterization of the antibodies, we made a pre-selection and with 20 candidates from mice, 11 candidates from rats, and 60 candidates from rabbits. The next step, having the sequences in hand and the binding data, we started already to do some in silico analysis. So we looked for developability and liability scores by using the ENPICOM's AIGX platform. And this table just shows a rough overview about the different parameters. And then we'll just briefly talk about the TAP score and the liability score. So the TAP score is the theoretical antibody profiler. And this compares variable sequences, the antibody variable sequences against 5 developability guidelines derived from clinical stage therapeutic antibodies. And the best score is 0, and the same holds true for the liability score, which is somehow self-explaining. It just is looking for potential amino acids that can create issues in comparison if you compare this to clinical stage antibodies. And as we can see, we found a number of good candidates. This table does not include all the antibodies. This is just a subset. But what we can see that we found very good antibody candidates, irrespective of the species and the immunization path that was used. Maybe just another comment to the AIGX platform. This also allows you to look exactly where these liabilities, which are basically within the amino acids or different features of different amino acids, by 3D structure models, you can exactly look where are these liabilities located. And this can be very helpful if you're looking for structural antibody target interactions. Okay, this is the one hand. On the one hand, we have the in silico data or first in silico data concerning developability and liability. However, when it comes to further characterizations such as binding kinetics or epitope binding, for this, of course, you need materials. So this is determined experimentally, and therefore you need purified, antibodies, either purified or as a crude supernatant. And if you have a lot of antibodies, which you can analyze, as it is the case for our RAGE panel of antibodies, you ideally have the capacity to do high throughput, small scale production of the antibodies. And luckily at Genovac, we have these capacities, so we can use small scale high throughput expression. And we did this in this case for the further downstream binding kinetics. And we can also do high throughput, small scale expression and purification. And these antibodies we used later for the epitope binding. And for further downstream, more quality related and functional assays, for example, we have also the option to do intermediate scale expression purification. Okay, so now having all the materials or the antibodies in hand for the further downstream characterization, meaning binding kinetics and epitope binding, we could move on to the characterization of the anti-RAGE antibodies. And for this characterization, you of course need the right equipment, and consequently, we used our Carterra LSA, a high throughput SPR system, which enables screening of a lot of different antibodies in one single experiment. So the print head of the instrument is able to print up to 684 different antibodies on one chip. And the good thing is you need really only small amounts of samples. So this is making you saving time and resources, of course. And then on these spotted antibodies, we can flow the analytes. For example, the protein for the binding kinetics in a one-on-many format, and you have then different cycles, and then you can analyze the data. So in general, we use the Carterra LSA core antibody we use the Carterra instrument for determining binding kinetics and affinities and epitope binding, and also we can do label-free quantitation of the antibodies. And as I mentioned, the good thing on the system is that in each case, you can use either crude supernatants or purified material. So in our initial kinetic determination experiments, we used the parameters which are indicated in this table on the left. So we used a protein AG surface chip to which the different antibodies are bound on the surface, of course. And we used a certain print density of 0.222 microgram per milliliter. And on the right-hand side, you see the workflow. So the first step is, of course, you have to capture the chip with the antibodies. Then you have to equilibrate the settings with buffer conditions. And then you flow the analyte, in this case, the purified RAGE ECD protein, onto this antibody surface. And if you see the binding of the protein to the antibodies, you will see an association signal. And then the next step is that you flush the cell again with buffer, and you will see the dissociation. And then this step, you repeat the cycle starting from establishing the baseline, then put the next concentration of the analyte. So in general, you have to start with the lowest concentration of the analyte, and then you move on consecutively with higher concentrations. So you don't regenerate this chip in this case, because you, of course, want to keep the antibodies on the surface. And this table summarizes already the results we got from these kinetic experiments. And we were really thrilled. We can say we were really thrilled by seeing these high affinities which derived from the rabbits. So we obtained single-digit picomolar affinities from the rabbits, but in general, we were able to obtain very high affinity antibodies also from mouse and rat. Another interesting aspect is that on average, we found the highest number of very high affinity antibodies in the genetic rabbit cohort. And these very, very high affinities is quite interesting how we finally really measured those, because initially these high affinities caused some, I will not say issues, but some extra experimental adjustments. And just we'll briefly want to touch on this. So as I mentioned, The initial binding kinetics were measured on a standard chip. And as we saw for the rabbit antibodies in the association phase, we saw very high linearity of the association sensogram signal part. And this is, not good, or we can say this is suspicious. And also together with the observed K-on rates, which were really high in this case. And this combined with this high linearity of the association phase, this is indicative of the fact that our measurements were mass transport limited. And so this is influenced by the so-called mass transport coefficient Km, which has to be above a certain level. And for these rabbits, we were always below these levels in most cases. So and if you observe these kind of mass transport limited kinetics, this in the end makes you measuring wrong affinities and values. So we had to do a second run, let's say like this, where we changed the parameters. And instead of using the standard chip, the Pro AG HC30M, which is more 3D hydrogel like chip, we changed to a more 2D planar chip, and we reduced the density of the antibodies. And this can help to limit the effects of these mass transport effects. And as we can see now, using these parameters, we see a much better curvature of the association force as we normally would expect it. And so by this, we can now be sure that we really measured the right K-on values and consequently the right, very high affinities for the rabbits. And just showing this graph again, illustrating that within the rabbits, we saw a much higher number of very high affinity antibodies when we used the genetic immunization in comparison for the protein immunization. So we were really happy to see these data. Okay, finally, we were also interested, of course, to look for the epitope binning, which also works very nicely with the Carterra instrument. And by the epitope binning, the epitope binning in general can serve as a surrogate for functional diversity characterization and can indicate potential functional classes of your different antibodies. Again, here on the left-hand side, the table shows the parameters that were used, and on the right side is the flow diagram of the experiment, which looks a bit different now. Of course, we have, again, a baseline. And in this case, the antibodies are covalently connected to the chip surface. If this is equilibrated, we first floated the analyte over these antibodies, which in this case is the protein antigen. So again, purified RAGE. And then you see the signal for the association of the analyte. And then in the second step, you flow your solution phase antibodies over the surface. And if you see binding of the antibody from the solution phase, you will see again association phase signal. And this means your two antibodies, your antibody pair, the immobilized and the solution antibodies bind simultaneously to the analyte, the protein, meaning that the antibodies recognize different epitopes on the target. And these antibodies will end up finally, or will be sorted into different bins. If you have a solution antibody flown over the surface that does not bind, this indicates that the binding site is already occupied by the immobilized antibody. So it's a blocking interaction. So this antibody recognizes the same epitope and so these two antibodies are in the same bin. Then again, you have to flush the surface with buffer. You see a dissociation signal. And in this case, you regenerate the chip, meaning in this case, choosing the right buffer conditions to flush away the solution phase antibodies and the analyte, the protein, and the immobilized antibodies are covalently fixed to the chip. So then you can start the new cycle by adding your next antibody, which you want to test against all the other antibodies. And so this is typical results. You see when you are doing epitope mapping, and in general, you receive a lot data, very complex data. But the nice thing is that Carterra provides really good software packages that facilitates the data analysis. And as you can see here on the left side, we have the heat map, where we have all the red indicated pairwise interactions. So on the vertical lines, we have the immobilized antibodies. And on the horizontal rows, we have the solution phase antibodies. And so each antibody was tested against each other antibody. And the red interactions indicate blocking antibodies. So these are antibodies binding to the same epitope. And the green ones are so-called sandwiching antibodies. This means they bind in parallel to the target antigen in parallel to the antibody that was on the immobilized chip surface. On the right-hand side, you see this network map, which indicates the different bins we found for these in this experiment for the RAGE antibodies. And there's a couple of things we can observe. So we have, let's say 3 clusters of very many antibodies with different bins. And we have 2 single antibodies, which are universal sandwiches. So this means they don't interact or they don't block any of the other antibodies. So these are probably antibodies recognizing a unique epitope. And in the middle, we see a control antibody, which is an anti-His antibody, which we had used as a control. So the analyte, the purified RAGE protein, contained a His tag. And so you would expect, and you need to see this, that this positive control antibody is a universal sandwicher. So as it does not or should not compete with any of the other target-specific antibodies, which indeed is the case here. The network maps, they can be applied in different ways. So one can also group the antibodies to different communities. So in the previous map, the bins were assigned to antibodies with identical competition behavior. But if you look for the community plots, you can put antibodies into communities which are show very similar competition profiles. So not identical, but very similar. And then you see that you get quite some big communities, and this gives you more information about the different epitopes and potential classes or functional classes of these antibodies. And if you recall the structure of the protein itself, the RAGE protein, you might recall that we had these 3 main domains, the variable and the two constant domains on the ACD. And it is somehow tempting to assign these different clusters of antibody groups to these 3 different target domains. although we cannot prove this because of course we don't have epitope mapping data in hand. But yeah, it looks like these clusters bind to the different major domains. Another interesting aspect is if you apply communities indicated by species, we can see, for example, that we have different communities. For example, this big one, which is exclusively rabbit, and we have other larger communities like this one or this one, which is exclusively rodent. So yeah, to us, this again indicates that yeah, including multiple species in such an immunization campaign, we are pretty sure that this increases the in chances for yeah, or to maximize the epitope coverage. And finally, at the end, I would like to summarize some key points, so which are delineated here in this my last slide. So immunization of multiple species combined with state-of-the-art single B-cell discovery maximizes the chances of discovering high specificity and high affinity antibodies. And another different important aspect was that the genetic immunization of the rabbits can result in yeah, target-specific antibodies with extremely high affinities in the single-digit picomolar affinity range. And finally, yeah, the Carterra LSA high-throughput SPR instrument enables high-throughput measurements of a lot or of a very high number of antibodies in parallel for binding kinetics and epitope characterization. And this, in general, provides you very valuable information, which is critical for the selection of therapeutic and diagnostic candidate antibodies, which you then further can process, for example, as we're dealing with wild-type animals, to humanization. And having said this, I'm very happy to hand over now to Piotr, who will talk about the ENPICOM platform and the humanization.
Thanks, Andreas, for the work. Indeed, I will be sharing our story about part 2, which is indeed very much about the candidates that we have just shown, how they have been discovered, how we have actually humanized those, and how we partnered up to do so. So we'll present on GenovacAI powered by ENPICOM, which is a deep learning solution and service for people looking for the state-of-the-art humanization service. Because this is taken directly from the Bio5 paper, which is the fact, basically summarized as the fact that although transgenic animals and display technologies have been greatly improved over the years, still loads of the antibodies are actually coming from mice or other model species, which require humanization. And traditional approaches are typically laborious and require a lot of knowledge. And that's some of the problems that people face. And also the Genovac team mentioned to us that they had similar problems. In fact, being that it was time consuming to do, for example, the grafting, relatively low throughput, and that very often also it actually results in loss of affinity, loss of binding. We already briefly mentioned other tools that one can use. However, also to set up those up, to maintain those, to get it all configured right, and then getting good results from it, it's time consuming, still relatively low throughput. And also most importantly, maybe, is that also with those tools, very often there is a lack of retention of the binding. So that's how we came to work together. We have done so for various years now, but for humanization specifically, we came together to set up a state-of-the-art service, which could include antibody discovery from Genovac, as we're presenting today. But you can also have already a molecule that needs humanization. So we have a model that does humanization, and Genovac then has the lab to screen those, to validate everything, and to even provide you with the materials. The great bit is that it's species agnostic. It's highly successful in actually retaining the binding affinity. And we optimize to clinically relevant humanist levels, which is of course key. On top of that, it's high throughput, which means that you can actually humanize directly post-discovery, making the process much quicker. Of course, it's secure, and all of this combined makes that it's also a very cost-effective solution. But maybe quickly introducing our company, ENPICOM. We've been in the business for about 10 years. We have, on the one hand, a biologics discovery software solution, the ENPICOM platform, which Genovac already mentioned a little bit, has been using. And on the other hand, we have our own machine learning assets and services. And this is what we focus on, basically. It's going from the early discovery phase, where you might have repertoires or libraries with billions of potential clones and candidates to select from, all the way up to the engineering to deliver then a couple of therapeutic candidates. And all of that, we, of course, aim for AI-aided research. And I'll tell a bit more about that in a bit, because the ENPICOM platform has everything there is that a scientist needs. So it's a very easy to use, intuitive interface that requires absolutely no coding, that allows you to process any type of sequencing data, whether it's Sanger, NGS, 10X, Oxford Nanopore, PacBio, etc. Process all the data with the gene annotations, the quality controls, and everything you would expect there, to the developability screening based on structural modeling, as Genovac already mentioned in the part 1 presentation, and of course, clustering, enrichment analysis. And as mentioned, everything is a very easy no-code interface, as you can see on the right top here, which allows scientists to really quickly and easily do their analysis and make decisions. However, it doesn't just stop there. So the platform offers this interface that directly in the background takes care of all the data management as well, with a very scalable system that can actually manage all your historical data, both the sequence data, and very good to mention as well is also the meta information. So any assay coming from the lab, if it's a thermostability, if it's a binding experiment, any information you can pull together with your sequence data, and that can scale up to billions of clones easily. And you have all the handles you need as a scientist to do your analysis. However, on top of that, with the rise of AI, especially in the biologics field, we noticed the need for a more extensive platform, and especially the collaboration between the data scientist and the lab scientist. That's why we also take care of the data scientists. We have their back, and with the platform, the data scientists can actually very easily retrieve data from the people working in discovery and engineering. Any sequence data, any lab data, retrieve what they call labeled data sets for model training. And also, of course, the later experiments. Everything can be fed into the data architecture and very easily retrieved by the data scientists, which is a very key task in their work in creating AI models. We provide the whole infrastructure for what is called MLOps, basically versioning, keeping track of models, training new models, and especially also deploying those models. Because one of the key problems in AI and biologics research is the effective use of AI models and making the actual research in the lab, making that aided by the AI models and doing better. And that's because the data scientists can very easily deploy new models that they've trained on the company's own data and deploy them in the interface for the scientist. You can directly include this in your antibody discovery or engineering research. And that brings that the investment on AI is actually much enhanced and much better. But what if you don't have your own ML team? ENPICOM, as I mentioned at the start, at ENPICOM, we have years of ML training. We've done various projects for pharmacy and biotechs alike. And we have, over the years, actually created our own machine learning assets. Part of that is the family of AIGX protein language models, which are basically very large machine learning models about what an antibody actually is. So how do you then use this? We have trained these on well-curated data sets. And subsequently, we can use these foundational models to train towards specific objectives. In this case, we used a humanization objective, and we have created the humanization layer for the model. If you then use this, you can take the input sequence, and as the output, we'll get a set of humanized variants. But AIGX can be further specialized to other tasks as well. So, for example, thermal stability or any other property. In this presentation, we're discussing the humanization and the campaign, and we'll get to the data in a bit. But to recap the objectives that we set for ourselves, is it should have a high success rate, reduced need for expressing many variants, is again much quicker, much more cost effective, and therefore a very great thing to have. On top of that, of course, we'll need to reach clinically relevant humanist levels, as we were aiming to make these into therapeutic molecules. Species technology agnostic to, as mentioned, Genovac has a wide variety of species, and we have made this model to be completely species agnostic. And also high throughputness to make sure that you can do the humanization earlier in the process and therefore skipping steps in research because you can do it all at the start real quick. Then to show a bit more on the data. So we have this humanization case study. We'll be taking the clones from the campaign that Andreas already explained in part 1. And we have actually been taking these. So this is a quick summary again. So we have taken 8 from the rats and 8 from the mice. They came from the Beacon, were then characterized with the Carterra LSA. And we've taken 8 of those of a genetically diverse data set for humanization. But first, let's discuss what we deem successful humanization criteria. So as we can see here, this is data from these publications. It's on anti-drug antibody response on molecules that made it to the clinic. And we see distributions on the left top on the human and the humanized molecules that made it to the clinic. If we look at the bottom plot, that's where we see the percentage of ADA. So, but the important bit is, what do we actually find a successful humanization? One of the key criteria was that we need clinically relevant humanist levels. And as you can see here, these are plots on ADA response, so anti-antibody responses. And as you can see, from 80 to 85%, that's where the range of molecules that made it to the clinic that have been humanized on traditional methods basically aim for. If we then move a bit further, we see that from 85, 90%, that's where we actually see most of them, as well as the human candidates where they start, the human therapeutics. But maybe more important is, where do we actually see a drop in the anti-drug response? We see that, as you can see in the bottom left, if we move from the 80 to 85, we still have quite significant ADA responses. Well, if you move to the right, the 85, 90% or 90 to 95% bins are the ones where you actually don't see a lot of drop in such responses anymore. So these would be, for us, the sweet spot of humanization, so that we reach certain the clinically relevant ranges. Then, of course, binding is key. We need to retain binding to do a good job, because if we can get to 100% human, but we lost all binding, there's still no good in the humanization. So what we did is that we defined a cutoff of either a 2-fold or a 5-fold of KD change from the parental antibody. And again, everything was measured using the Carterra LSA platform. And then what does this actually look like? So what you're seeing here is the success rates that we achieved with the ENPICOM model, with the AIGX model. So on the top, you see what is the success rate if we take a fold change cutoff of 2, which is quite strict, I would say. And at the bottom, you see the same data, but with a cutoff of a 5-fold difference in KD between the parental and the humanized variant. All of this is, as mentioned, for the 16 parental, 8 mouse, 8 rat antibodies. To put this into perspective, we found that we should do a benchmark. We wanted to compare how our model performs, how it actually compares against other tools that would be out there. And we took the BioPhi model as a benchmark. This is what we are getting with the BioPhi platform. So if you're looking at the left, we see there's still reasonable amounts in the 80% germline identity, that's human germline identity. But the moment we move to more higher humanist levels, we start to notice significantly drop in binding retention using BioPhi. And that's really where the AIGX model starts to make the big difference. Then on top of that, we have looked at from everything that was binding and all the clones that did very well in the humanization campaign. If we take the top 25, what actually happens? And you see that the ENPICOM model also creates significantly better binders. So from the top 25 binders, we have 23 of them are actually generated with our model. And by creating better binders, higher success rates, the great thing you have is that you actually need to express less candidates. And if you're expressing less candidates, it saves both time and cost. And in the end, that translates, of course, to a more effective campaign. Maybe one thing, is of course the species agnostic part to the model. We want to be sure that we can humanize any species with the model. And if you remember, it was already mentioned in part 1, for this campaign on RAGE, we generated very great rabbit antibodies. These rabbit antibodies show to have low picomolar affinity. And we set ourselves up for a challenge. So we have extremely tight binders that with the slightest change and slightest problem, the binding will be significantly reduced. And on top of that, rabbits are known to be much further away from human sequences. As you can see here on the right, all the way to the right, we see the distribution of human antibodies. And if you move down, you see that with rhesus, with rats, with chimeras and mice. And then on the left bottom, we see where rabbits are. So the further away we are from human, obviously, the more we have to mutate to get to reasonable humanness. And we took 2 very strong binders from the rabbits, and we both successfully humanized those to at least 85% germline identity, human germline identity. And we wanted, and we have achieved to retain the low picomolar affinity. So as you can see here, we have stayed at like 132, 346 picomolar affinity for a fully humanized candidate that actually made it more than 85 germline identity. Then on top of that, this is data that we did in a campaign some time ago. We have only kept improving the model, and we have found very great improvements in both success as well as binding retention, because we have created a pair chain aware optimization, which basically means that the model understands the combination of the heavy and light chain as being one molecule that can then be humanized in the same go. And therefore, we don't need anymore the combination of different heavies and light chains, because the model will do it for us and just suggest a combined heavy light chain humanized clone. And on top of that, we have looked further into protecting key residues, which again created better binding retention. And in the future, we can do more multi-objective optimizations to also co-optimize for other properties. So in summary, with the GenovacAI humanization service that's powered by ENPICOM and our model, we have created deep learning aided service for next generation humanization. So if Genovac did the antibody discovery, it can go directly into a humanization service. If you have your own molecules already, that's also perfectly fine. Then with the AIGX ENPICOM model, we create humanized variants of your parental sequence. And from there, Genovac has a state-of-the-art lab to do the screening, the validation, and can even deliver to you the product and the antibody being expressed. And in a recap, all of this is species agnostic. It's highly successful in retaining the binding affinity. We reach the clinically relevant humanist levels, and we can do it in high throughput, which means that you can do many of them. You have to express low numbers of clones to get to a success, and therefore it makes it really cost effective. With that, I want to thank you all for your time. If there's any questions, we would be happy to take them right now, but also feel free to shoot us an email later if any question comes up. Thanks a bunch. And with this, I want to give the word back to Elizabeth.
Thank you very much, Piotr and Andreas. We do have quite a few questions, but only a couple minutes to address them. So I will start with our first question from Stephen. Do you have any thoughts why it was so challenging previously to get good antibodies to the RAGE antigen using protein immunization?
Okay, maybe I take this question. Thank you, Stephen, for the question. So, yeah, per se, I think it's obvious that the RAGE protein, although it has a type 1 protein structure, might not be a very easy target. So that's why we opt for a Beacon screening from the beginning. Per se, I think the Federal Lab has performed mainly hybridoma attempts for the screening. And I think just that one reason is that the single B cell screening simply outperforms the hybridoma. And also, I'm not quite sure which protein was used in their attempts, but we have produced the RAGE protein ourselves in our protein production facility. And we normally just produce very high quality protein, and we use this for our campaign. So I think, this is maybe a second reason why, but I think the first reason is the more probable one, that just the single B screening, B cell screening, is the more suitable approach in this case, or was the better choice in this case.
All right, thank you. We have a request for clarification from Reza. I thought it was only 384 antibodies per chip. Yeah, thank you, Reza, for pointing on that. So this was just, I'm sorry, I was just misspoken. But however, if you, of course, you can regenerate the chip, and then you can load a second set of another 384 antibodies to the chip. But I just, misspoke the correct number. Sorry for that. Okay, no problem. Our next question is from Anitha. With the AIGX platform for the TAP score, did you do repertoire sequencing using NGS or Sanger?
Yeah, Andreas, do you want to take it? Yeah.
You can take as well, Piotr. So in this
doesn't matter. We've seen the data both. In this case, I think it was all Sanger sequencing, but both the Genovac lab and the AIGX platform are more than capable of indeed also generating the NGS 10x and Illumina sequencing. So for this campaign, it was Sanger, but there's also plenty of NGS going on.
All right, and one more question. The bioinformatics toolkit, is that available free or on GitHub?
No, unfortunately, it's not. That's actually ENPICOM's main product, I would say. We work very hard to provide this intuitive user interface. And it's being used by pharmas, biotech heroes around the world. So unfortunately, it's not free to use on GitHub, but yeah, please reach out and we'll be happy to see what we can do there.
All right, I see we've reached the end of our time. So thank you both to our presenters, Andreas Weisse and Piotr van Rijssel, to our sponsors, Carterra, ENPICOM, and Genovac. So with that, on behalf of Cambridge Innovation Institute's Global Web Symposia series, thank you all so very much for coming and have a great rest of your day.